At the world's most secure labs in the Nevada desert, researchers blasted, froze, and starved microscopic creatures in experiments designed to reveal life's ultimate limits. They expected to find a breaking point, but instead they watched tardigrades survive what should have been instant death. Temperatures colder than Pluto, radiation strong enough to sterilize a nuclear blast zone, even the hostile emptiness of space. None of it works. If Nevada's best can't kill them, what does that mean for our understanding of life? And why are these labs so determined to try? The answer rewrites everything we thought we knew about survival. Nevada's research footprint stretches across more than 40 specialized laboratories, with the Desert Research Institute alone accounting for a sprawling network that covers everything from atmospheric science to radiation exposure simulations. In 2025, the Desert Research Institute reported $59 million in sponsored research, a figure that rivals many state universities. The numbers suggest a place built for the edge of possibility, a region primed for experiments that test the boundaries of life. Access to these facilities is tightly regulated. The Nevada National Security Site, once the proving ground for atomic weapons, now requires multiple layers of clearance just to approach the perimeter. Permits stack up: environmental, radiological, even archaeological. The process is labyrinthine, designed to keep secrets in and prying eyes out. Yet, for all the infrastructure and funding, something is missing. The expected trail of paperwork, grant awards, published protocols, archived experiment logs, vanishes where it should be most visible. State registries show no record of contracts for extremophile research. Patent offices come up empty for biomolecular stabilization linked to Nevada labs. Even the internal newsletters, usually quick to trumpet new discoveries, remain silent on any work involving tardigrades. This silence is striking. Audits of public grant databases, including the National Institutes of Health, the National Science Foundation, and the Department of Energy, reveal 0 awards tied to tardigrade studies in the state. No small business innovation research contracts, no startup incorporations, no press releases announcing breakthroughs in desiccation biology. The Desert Research Institute's own annual reports highlight climate, water, and archaeological wins, but extremophile biology is nowhere to be found. The absence is not just a gap, it is a void, one that grows deeper the more resources are tallied. For every $1 million spent, for every lab opened, there is an equivalent silence in the scientific record. Even the most basic step, filing a Freedom of Information Act request for unclassified contracts, returns nothing but redacted forms or polite denials. This disconnect between massive capability and missing documentation plants a seed of doubt. Why would a state with the infrastructure to study the limits of life leave no trace in the archives? The mystery is not just about what Nevada's labs can do. It is about why, with so much power at their fingertips, the official story contains so little. Nothing in Nevada's official records hints at what these microscopic creatures can actually endure. But the numbers, verified across decades of global research, are so staggering they sound like science fiction. Tardigrades have survived temperatures as low as -458 degrees Fahrenheit, just a hair above absolute zero, and as high as 300 degrees Fahrenheit, hotter than boiling water. They have shrugged off blasts of radiation 1,000 times deadlier than the dose that would kill a human, withstanding up to 5,000 grays of gamma rays in controlled lab tests. In the absence of water, they dry out completely, curling into a cryptobiotic state where metabolism nearly halts. Decades can pass. Then, with a single drop of moisture, they reawaken and walk away as if nothing happened. The vacuum of space, with its crushing cold cosmic rays and ultraviolet bombardment, has failed to finish them. In 2007, 3,000 tardigrades were launched into low Earth orbit aboard the Photon M3 mission. For 12 days they floated in the void, exposed to the full fury of solar radiation and the emptiness of space. 68% survived rehydration back on Earth. Some even laid eggs that hatched healthy offspring. In other experiments, tardigrades endured pressures 6,000 times greater than our atmosphere, far beyond the deepest ocean trenches. Radiation, dehydration, freezing, boiling, vacuum, and crushing pressure. None of these alone or in combination have managed to erase a tardigrade's spark. The secret lies in cryptobiosis, a reversible state that rewrites the rules of survival. When conditions turn lethal, tardigrades shed almost all their water, fold in on themselves, and deploy a molecular arsenal. Trehalose sugars to glass their cells, unique proteins to shield DNA, and repair systems that leap into action the moment life resumes. Scientists have revived specimens after 30 years of dormancy, watching them spring back to life in a matter of minutes. These numbers are not theoretical. They are the benchmark, the line in the sand that every new experiment must face. For Nevada's researchers, the challenge is clear and daunting. To claim a breakthrough, they would have to surpass a creature that has already survived the extremes of the universe. The question is not just how to kill a tardigrade, but whether it is possible at all. Rachel Schulte's name never appeared in the official grant ledgers or the university's list of principal investigators. Yet among those who study life at its outermost limits, her fieldwork is quietly well known. In the spring of 2023, Schulte traced a path along the Sierra Nevadas high-altitude ridgelines, collecting lichen samples from elevations above 9,000 feet all the way down to the outskirts of Fresno. She became an expert at coaxing tardigrades from their cryptic refuges, mastering extraction techniques that left even seasoned biologists impressed. Her notebooks filled with catalogue numbers, microhabitat notes, and the subtle differences in water content that could mean the difference between a living tern and a desiccated shell. For Schulte, the search was never just about cataloguing species. It was about pressing up against the boundaries of what life could endure and finding out if those boundaries really existed. Dr. Marcus Hale, by contrast, operated at the institutional core, a director tasked with translating Nevada's research power into measurable results. His mandate was clear: design experiments that would push tardigrades through a series of escalating stress tiers, each more severe than the last. Extraction transects mapped the journey from pristine alpine mosses to the irradiated soils of the desert lowlands. Hale's background in environmental stress physiology made him a natural fit for the job, but it was his willingness to challenge accepted limits that set the tone for the lab. He demanded protocols that would not just test, but redefine the known thresholds for survival. The goal was not to replicate what had already been done in European or Japanese labs, but to find the point where even tardigrades faltered. What united Schulte and Hale was a restless curiosity, sharpened by the knowledge that every experiment ran up against a wall built by the creatures themselves. Tardigrades had already survived the vacuum of space, shrugged off gamma radiation, and outlasted decades in a state of suspended animation. For Schulte, each new sample was a chance to witness resilience in miniature. For Hale, every data point was a challenge to the dogma that all life has limits. Their collaboration transformed routine fieldwork into a high-stakes investigation, one that blurred the line between the methodical and the audacious. Inside the lab, the atmosphere reflected this duality. Schulte's careful labeling and meticulous sample prep met Hale's drive for ever harsher test conditions. Extraction trays lined up beside radiation chambers and cryogenic freezers, each station a waypoint on the journey to the edge of biological possibility. The stakes were no longer theoretical. With every trial, the question became more urgent. Could Nevada's scientists find a way to do what space, radiation, and time itself had failed to accomplish? The search for that answer would demand not just technical skill, but a willingness to confront the unknown, one experiment at a time. The experiment began with a protocol designed to erase any margin for error. Tardigrades collected from high-altitude lichen were placed into a chamber where temperature, radiation, and atmospheric pressure could be dialed up or down in precise increments. The plan was to subject each specimen to a gauntlet of stressors, not in isolation, but layered, freezing to near absolute zero, followed by pulses of gamma radiation, then a sudden drop to vacuum. This was no ordinary trial. Every variable was logged in real time. Each step cross-referenced against survival data from the Photon M3 mission, where 68% of tardigrades had revived after 12 days in orbit. The Nevada team wanted to push beyond those numbers, to find the point where even the most resilient fell silent. Dr. Marcus Hale kept watch from the control console, eyes fixed on the digital readout as the chamber's internal temperature plummeted. Rachel Schulte hovered nearby, her field notebook open, pen poised above the margin. She recorded the exact sequence, minus 272 degrees Celsius, 5,000 grays of gamma radiation. Atmospheric pressure dropped to a near-perfect vacuum. The chamber's sensors tracked every heartbeat, if there had been any to track. After the last cycle, the system registered flatlines across all biological markers. The specimens, curled into their cryptobiotic tons, showed no visible signs of life. For a moment, the lab fell quiet. The revival protocol began with a slow drip of distilled water, rehydrating the samples one by one. Minutes passed. Then, as the clock ticked past the 30-minute mark, movement flickered under the microscope. One tardigrade unfurled, legs twitching. Another extended its snout, probing the glass. Within an hour, more than half of the test group had revived. Schulte's hand shook as she wrote the timestamp in her notebook. In the margin, she scribbled a single word: impossible. The data log captured the sequence: temperature minimum reached at 342, radiation pulse at 344, vacuum sustained for 12 minutes, rehydration initiated at 4:03. First signs of movement occurred at 4:34. Out of the original set, 68% revived, matching the photon M3 precedent almost exactly. The numbers refused to budge, no matter how the parameters shifted. Every attempt to push the system further yielded the same result: tardigrades that should have been dead, returning to life with no detectable loss of function. For Hale, the outcome was more than a failed hypothesis. It was a rupture in the logic of the experiment itself. The margin for error had been eliminated, yet the creatures survived. The notebook, now heavy with annotations and timestamps, became a record not of success or failure, but of disbelief. The boundary between life and death, so clear in every other organism, dissolved in the face of this evidence. The search for an explanation could no longer rest on technical limits. Something deeper was at play, a molecular resilience that defied every rule the lab had set out to test. Tardigrades owe their survival to a series of molecular maneuvers that begin the moment danger closes in. At the heart of this transformation is the ton, a compact, glassy state that turns a plump, eight-legged animal into a microscopic capsule. Water loss is the first trigger. As cells dry out, tardigrades produce unique proteins called CAHs, which stands for cytoplasmic abundant heat-soluble proteins. These proteins act like molecular scaffolds, holding the cell's delicate machinery in place while the rest of the organism shrivels. The cytoplasm thickens, slowing down every chemical reaction to a crawl. Instead of collapsing, membranes and enzymes are locked in a protective gel. Inside the nucleus, another player steps in. The DSUP protein, which stands for damage suppressor, wraps itself around DNA strands like a shield. This barrier absorbs the impact of gamma rays and cosmic radiation, preventing the double-strand breaks that would shatter the genetic code in any other animal. Researchers watching fluorescence-tagged tardigrade day under stress have seen DSUP coat the chromosomes, blocking the formation of lethal fragments even as radiation levels spike. The process doesn't stop with proteins. As dehydration deepens, tardigrades switch on a suite of genes that drive vitrification. Sugars like trehalose flood the cell, replacing lost water and forming a glassy matrix. This vitrified state halts metabolism almost completely. Oxygen is not needed, waste stops accumulating, and the risk of ice crystal damage vanishes. The animal can remain in this suspended state for years, waiting for conditions to improve. Yet the exact signal that flips the switch to cryptobiosis remains a mystery. Scientists have identified dozens of genes that ramp up during stress, but the master control, what tells a tardigrade to enter the turn, has yet to be found. Every attempt to push the system to failure, to find the moment when these defenses give out, has ended with revival. The molecular choreography plays out the same way each time. Proteins assemble, DNA shields deploy, and the tone endures. For those in the lab, the spectacle is almost hypnotic, a living cell freezing itself in time, then waking up as if nothing happened. Organ preservation used to mean a race against time. Now, researchers are looking to tardigrades for answers. In 2024, a team at the University of Wyoming demonstrated that proteins borrowed from these microscopic animals could keep mammalian organs viable at room temperature for days. This advance could transform transplant medicine. By mimicking the vitrification process that lets tardigrades survive drying, scientists have begun to stabilize blood and tissues without refrigeration, opening the door to safer, more equitable care. The implications do not end with medicine. When the Italian space agency BioKiss mission exposed tardigrades to direct sunlight in orbit in 2011, the animals returned to Earth alive. Their DNA, shielded by ZUP proteins and glass-like sugars, showed minimal damage even after days of unfiltered solar radiation. For those planning long-duration spaceflight, these results are more than a curiosity. They are a blueprint. If human cells could be engineered with similar defenses, radiation exposure on Mars or in deep space missions might become a manageable risk, not a fatal one. Yet as the promise of tardigrade-inspired technology grows, so do the questions. The same resilience that makes these animals valuable as models also blurs the line between scientific progress and ethical restraint. Organisms once considered too simple for concern are now at the center of debates about the limits of experimentation. Should there be boundaries to how far we push life in the name of discovery? Is it right to subject even the hardiest creatures to conditions that would obliterate anything else? Dr. Evelyn Boothby, who leads the Wyoming protein research, has called for an international review board to oversee extremophile studies. She argues that transparency and oversight are essential as research leaves the petri dish and enters clinical trials or space agencies' toolkits. In her words, the goal is not just to see what life can survive, but to decide what we should ask of it. The debate is no longer theoretical. As tardigrade-derived proteins move from lab benches to hospital wards and astronaut suits, society must weigh the promise of survival against the cost of pushing living systems to their breaking point. The question is not just what tardigrades can endure, but what we are willing to demand of life itself. Today, tardigrades remain the only known animals to survive direct exposure to outer space, challenging everything we assume about life's fragility. As scientists race to engineer resilience for humanity's future, these microscopic survivors redefine what is possible for biology on Earth, Mars, or beyond. The real question is not where the limits are. It is whether we are ready to follow where life, in its most unbreakable form, leads next.